Thank you for the introduction. I'm Xiaoyue, and today I'm going to talk about smoothing functions for data-driven evolutionary models of multi-domain protein architectures. So in this talk, I'm going to introduce multi-domain proteins and how domain architectures capture modular composition, and then I'm going to introduce the domain bigram model and smoothing methods for the bigram model. So what are multi-domain proteins? Multi-domain proteins are mosaics of structural or functional elements called domains, and domains are sequence fragments that encode a distinct structure and/or function. And the domains are found in otherwise unrelated proteins, and they fold independently in many different contexts. And for example, in this MPP6 protein, there are 5 domains that fold independently on their own. Um, in this 3D visualization. Um, And multi-domain families are encoded by genes that share common ancestry. And here I'm showing you an example of the McGurk family, and they have similar domain content but with variation. And the modularity of multi-domain protein structure is intimately related to the modularity of their functions. For example, these 4 domains in the cask protein each binds to different things, and this makes the cask protein function as a scaffolding protein that groups these other proteins together. And this is our mental model of how multi-domain architectures evolve. We model them as with gain and loss events of domains. And even though there are various molecular mechanisms that can lead to domain gains and losses. And a multi-domain protein can be represented by its domain architecture, which is the sequence of domains in antecedent terminal order. And in the mathematical notation, we add start and end symbols. And the tokens in this string are from an alphabet of all domain families represented in the genome. And for example, the size of the human domain alphabet is approximately 1100. And if you think of the amino acid sequence as a long string over a small alphabet, then the domain architecture is a short string over a much bigger alphabet. And the DA representation, the domain architecture representation, is an abstraction. And this abstraction throws out a lot of information, but it allows us to focus on the evolution and function of the modular composition of proteins. And now I have introduced multi-domain proteins and their domain architectures, and I will now introduce the bigram model and smoothing methods for bigram models. So examination of domain architectures in primate genomes shows that we see many fewer domain combinations than expected by chance. And this suggests that domain adjacency and occurrence are highly constrained. And another striking example is that the 2 most common primate domains did never co-occur in the same protein, while we expect them to co-occur many times given their frequencies. And one way to examine these constraints on multi-domain proteins is to consider a bigram model of domain architectures. And in a bigram model, the probability of a domain architecture is the product of conditional probabilities. This is looking from antecedent terminal order or left to right. What is the probability of observing the next domain given the previous one? And the conditional probabilities are estimated from bigram frequencies in genuine data. And I will use C to denote counts of domain unigrams and bigrams. And here, PDJ given DI is essentially the MLE estimate of the conditional probability, but I will simply use small p throughout this talk when I'm doing the calculations with genuine data. And since the bigram model gives the probability of a domain architecture, we can use it to do a lot of things. We can sample strings from bigram frequencies, and we can simulate the dynamics of domain gains and losses with MCMC based on this model. And another work also did species tree reconstruction based on bigram composition. And thinking about domain architectures in terms of bigrams is a simple but very helpful way for us to examine the universe of domain architectures and probe the constraints. And ultimately, it helps us understand the design rules of multi-domain architectures. And for example, when we compare bigram frequencies to unigram frequencies, we see that domain pairs differ substantially from the count of domain pairs differ substantially from what we would expect by chance, given the marginal probabilities. And the problem with bigram data is that it is very sparse. And as I've discussed, we only observe a tiny fraction of all possible bigrams. And so why do you see so many zeros in the bigram matrix? Some bigrams and therefore domain architectures, they are disfavored by evolution, and so we don't see them together. And also there can be incomplete sequencing or annotation error. And sometimes the combinations, the domain bigrams are actually viable, but they're not yet generated and tested by evolution. And so we need a smoothing method to fill in the zero entries in the bigram count matrix. It is partly needed by the math of the models, but it should also encode our hypotheses on the biological reasons why we don't observe something. And also selecting an appropriate smoothing technique is also a problem that arises in many other applications, not just modeling multi-domain architectures. So many bioinformatic applications use additive smoothing, including us. Here I'll be using C hat to indicate the adjusted bigram count after smoothing. And so for the additive smoothing method, we add a small pseudocount psi to all possible bigrams. And this way, as psi gets really large, all bigrams will have the same probability. So essentially, this method interpolates between observed bigram frequency and a uniform distribution. And a question is, we don't know how to pick a psi because the value is just a number and it doesn't carry much intuition. And so we come up with a second smoothing method, which we call L2 minimization. And here we choose a number alpha, which explicitly encodes a measure of data completeness. And we add counts to unseen bigrams. And probability mass-wise, we assign 1 minus alpha probability to unobserved bigrams. And with this method, we estimate the conditional probability by minimizing the weighted deviation of the conditional probability to the observed marginal unigram frequency. And here in the equation of the L2 minimization method, it is written in terms of adjusted counts. But if you consider probabilities, then as alpha goes to 0, so as we assume that the data is more incomplete, the probability of a bigram is proportional to the product of unigram frequencies. And therefore, this method does smoothing by moving the bigram distribution towards a unigram model where domains are drawn independently. And so comparing additive smoothing and L2 minimization, additive smoothing moves the bigram distribution to a uniform distribution, whereas L2 minimization moves it to a unigram model. And in additive smoothing, the parameter doesn't explicitly encode anything, but parameter explicitly encodes our assumption of data incompleteness. So to test, we did this in human. We had human domain annotations from superfamily, and there are over 5,000 unique domain architectures, not a lot. And to give you an idea of the scale of a problem, the average domain architecture length is 5 and median is 3. And there are over 1,100 unique domains. And to mimic incompleteness of the data, we took the genuine data and we randomly throw out 10% of observed domain architectures. And we use the 90% left to train the bigram model. And we assess impact of the two smoothing methods. And we measure a few things, and I'll discuss these. And in order to make an apple-to-apple comparison between the two smoothing methods, we calibrate the effective pseudocount, which is the amount of change made to bigram counts. And so the first question I ask is, how much does smoothing distort bigram frequencies? Obviously, you add a lot of counts to unseen bigrams, and the number of unseen bigrams exceeds the number of seen bigrams. And so here, I measured the error in bigram frequency to the genuine. And well, since you calibrate the effective pseudo counts, the amount of change is not too different between the two methods. And uh, whereas you fix the amount of probability mass moved, the two methods can move different bigram frequencies to different other bigram frequencies, and so there's a change. There's difference in error when the effective pseudo count is quite large. And then we also consider relative entropy as a measure of constraints, and this is essentially telling you how much information gain do you have when you consider bigram over unigram. And when the relative entropy approaches zero, that means the model is essentially very close to a unigram model. And so as the effective pseudo count is big for L2 minimization, the relative entropy drops to even below zero. And then we ask, how does smooth bigram model fit unseen data? And to answer this, we consider the log likelihood of the 10% domain architectures that we throw out. And so as when you start with a small effective pseudo count, L2 minimization does better than additive smoothing, because I think at this point you're handling the unigram model parts of the bigram model. And as the L2 minimization, well, as your assumption of the data incompleteness goes to unreasonably small, unreasonably high level of incompleteness, then the unigram model dominates, and you're no longer explaining the domain architectures that you observe. And so right now we're also considering other alternative smoothing methods, and the rationale is that smoothing should depend on domain frequency, but the observations for common domains are very likely true observations, and so we should trust the frequent domain pair counts more. And at the same time, we want to interpolate between observed bigram distribution and the unigram model. And up to now, I've only shown you effects of the smoothing methods on the numbers. And we actually ultimately want to look at effects of smoothing methods on biological properties of sample domain architectures. Right? We want to test with domain combinations known to be viable or known to not be viable. For example, DNA binding domains, they cannot co-occur with extracellular specific domains. And also we want to consider summary stats of domain architecture samples, for example, length distribution. As a starter, I have some examples with the additive smoothing. So here, as you increase, so here I'm showing you distribution of length of sample domain architectures. And as you increase your pseudo counts, you have exponential growth in the length distribution. Well, you won't be near the elbow anyway as you select your pseudo count, but this is a sanity check. So yeah, that was it. And I would like to thank the Duran Lab. Akshat, he did a lot of experiments. I only showed a few. And I want to thank Dr. Gautam Iyer, the mathematician, and thank our funding. And thank you all for listening.
Okay. Thanks, Xiaoyue. So we don't have a ton of time for questions. I'm going to ask a quick one while Dr. Patrick Bradley comes up. So my quick question is, so first of all, very impressive work. I'm wondering if there are barriers to using this beyond species in humans. So for example, if you build a bigram model based on the human proteome, do you think it's going to work similarly in the mouse proteome or other vertebrate proteomes, or even if you go further out evolutionarily?
Well, so I think if you build one bigram model in one species and you test its performance in other species, that's a way of asking whether there are different design rules in different species, different lineages. And so I guess for very closely related species, the design rules should be similar. We've tested this in primate and mouse. And so, but I would be curious to see how a model in one species performs in another species.
Okay,
Thank
thanks a
you.
lot.